All right, guys, this is the video over 1.4 membrane transport understandings. Just to say, you're going to need to know the difference between all of the passive transport and the active transport. I'll let you read through these. These are pretty easy to understand. Endocytosis and exocytosis. Endocytosis is used to bring big things into cells. And if it's big, you're going to need to use energy, which we call ATP. The main thing you need to remember is that endo means inside the cell and exo means outside the cell. And I don't have to read these to you because you can copy these notes in for yourself. But in this picture, we have large particles too big to go through the membrane itself. So when they get to the membrane forms around them and it forms a vesicle. A vesicle is basically a compartment surrounded by its own membrane. And it gets that membrane from the cell's membrane itself. It just kind of pinches off and it goes inside the cell. Exocytosis is the opposite. So we've got a vesicle here and it fuses with the cell membrane, releasing its contents to the outside. An example of this is like protein insulin. Insulin's made, you've got the insulin gene in the nucleus in your DNA. You make an RNA copy of that insulin gene. The RNA copy leaves the nucleus, goes through a nuclear pore or a hole into the rough endoplasmic reticulum. It's rough because it's got ribosomes on it. That RNA finds a ribosome on the rough ER and it gets made into insulin. The insulin then goes into the Golgi apparatus, which is just a sac of membranes, where that Golgi packages it and sorts it and ships it out in vesicles. And so then we see this vesicle here. Imagine these pink dots are insulin. This vesicle goes up to the top of the cell, it fuses the membrane, and it releases the insulin. Okay, passive transport. In all of these, we are going from high concentration to low concentration. That is the natural movement of things. Because molecules vibrate, they will bounce around and they will diffuse until they're equally spread out. So simple diffusion, these things can pass between phospholipids. These are only small things like carbon dioxide, which has 3 atoms, and oxygen, which has 2 atoms, O2. So simple diffusion is for things like gases that just can slip right in between until there's equal amounts on both sides. Facilitated diffusion, these are things like ions and sugars and things that are too big to slip between the phospholipid molecules, so they need a protein channel. It's kind of like a tube or a sieve. So the facilitation is done by the protein. It is still going from high to low, so it goes until there's an equal amount on both sides of the cell membrane. And then osmosis is the net movement of water from regions with low solute concentration to high solute concentration. What that means? Water loves solute, so it's going to go wherever the solute is. I like to think of salt. So like in an IV in a hospital, when they give you an IV bag, they can't just give you straight water. It's got to measure the same solute as your cell solute. So the same amount of salt in the bag has to be the same amount of salt in your cells. And I think it's 0.5%, but you don't need to know that. So isotonic, that's when the solution solute, like that IV bag, is equal to the cell solute concentration, that 0.5%. Okay, so then the cell's happy, everybody's happy if it's isotonic. Hypertonic, that's too much salt. Hyper means high. So high solute solution. If you put too much salt into your IV, remember water loves to go where the salt is. So the water's going to leave your cells. It's going to go outside the cells to join that salt, and it's going to cause your cells to shrink and die. Now, hypotonic, if it's too low solute, like if it was just straight water in that IV bag, remember, water loves to go where the salt is. So if there's no salt in the IV bag, all that water is going to rush into your cells and swell it up and burst. So it has to be isotonic. Now, a little trick to remember, the hypo has an O in it. And if you think of the O as a swelling cell getting ready to burst, that might help you. All right, we'll see you in part 2.